This recording is one of the recordings that are part of the lectures covering the circulation, and this one will specifically look at the types of blood vessels. So we'll start with looking at blood as it leaves the heart, and it's actually going to leave the heart via arteries. So arteries carry blood away from the heart. So we're going to look at the structure of different arteries, and then we're actually going to be kind of going in this order as we come along. Okay, so let's start with your elastic arteries. So elastic arteries could be the aorta if it's coming from the left side of the heart. It would be the pulmonary trunk, for example, which would be coming from the right side of the heart. They are strong. They're thicker walled than, say, corresponding veins. The reason why they're thicker walled is they have to withstand higher pressures. So if you look, just kind of look at, compare this artery versus this vein, you see this is much thicker than the vein because they have to withstand higher pressures. Now, the arteries have three distinct layers. You've got your tunica interna, which surrounds the lumen. It has the endothelium. So you have the tunica, some textbooks will call it the tunica intima. It's got a basement membrane, and elastic arteries also have an internal elastic membrane or internal elastic lamina. That's going to help arteries in general, to be able to return to their original shape, despite the fact that they have all this blood that's pushing up against the sides of the walls. Now, the tunica media is the thickest layer in all arteries. It has smooth muscle and elastic fibers. So you see here, see the smooth muscle here? It's thicker than you see in corresponding veins. It also has a tunica externa, also referred to as tunica adventitia. The outermost layer, it has things like elastic fibers, some collagen fibers. They're more for kind of adhering that blood vessel to surrounding tissues. It's one of the things that's involved in. Now, the arteries in general carry blood under very high pressure. Now, not all arteries, lots of times people memorize, oh, arteries have a lot of oxygen and low CO2. Now remember, arteries carry blood away from the heart. Pulmonary arteries are taking blood from the heart up to the lungs where it's going to pick up oxygen. So pulmonary arteries are not well oxygenated, but I do admit all the rest of the arteries are. Okay, so but don't automatically memorize arteries have high oxygen, because they don't. Now we start with this elastic arteries, which have a lot of elastin fibers, and they act as secondary pumps. So they're going to be able to allow blood to continuously flow through the circulation even as the ventricles are starting to relax. Now that blood travels from those muscular arteries the elastic arteries to muscular arteries. They're more distribution arteries. They're just distributing that blood to the various organs. They have, again, a lot of smooth muscle in those, more than the elastic arteries themselves. Now then they go to arterioles. So you see arterioles are a little bit smaller. Now arterioles are like technically very small arteries, but they deliver blood to the capillary beds where the exchange will take place. Now, there actually are more than one type of arteriole. We have resistance arterioles and true arterioles. True arterioles are the ones that specifically are delivering that blood to the capillary bed. Resistance arterioles, they give that name because these play a very important role in regulating blood flow to the capillaries. They're a primary site of vascular resistance. They're going to be important in regulating blood pressure in blood flow. So the smooth muscle of those arterioles are real important, specifically when you affect the contractility of the smooth muscle arterioles. That will have a major role in regulating your blood pressure. But arterioles, please write this down because we're going to encounter this again at some point. Arterioles, specifically resistance arterioles, big thing that they're responsible for is reducing something called pulse pressure and reducing mean arterial pressure. When blood hits those capillaries, capillaries are very thin-walled. I don't want pressure too high when it gets there because it could damage them. So it's important that those arterioles kind of reduce that blood pressure enough so we don't blow out those capillaries. So it's going to be very important. Now I want to look at capillaries, and we have different types of capillaries. There's the smallest vessels, they're very thin-walled, and we have different types. Now they're going to connect the arterioles to the venules, and the capillaries are where exchange of things will take place. Gas exchange, going to be delivering nutrients, picking up various waste products. Now we do have three types. The first type is what you see on the far left, is a continuous capillary. Continuous capillaries have an intact basement membrane, and the clefts that we see between individual cells, they're very, very tight. There's not a lot of space for things to get through. So remember, membranes being, the composition they are is if you're non-polar, you can get through. But if you're polar or hydrophilic, unless you have some sort of transport protein, it's not going to allow it through. So it's going to affect the permeability of that blood vessel to certain molecules. Well, if you have a gap in there, yes, things can sneak through, but if you don't, the only things you're going to get through are things that are very lipid soluble, like gases and very small ions, very small molecules. So a continuous capillary, of all the three types, this is the least permeable. This is also the most common type of capillary. So you see places that we'll find it. It's like in muscle, in the lungs, and in the brain. You'll find it also in like connective tissues. The second type is called fenestrated. Fenestra is a word that means windows. So you'll notice we still have an intact basement membrane, but you see these kind of little holes that we have in the endothelium. And so this allows little larger things to get through. So let's think about why we'd want that based on where we'd find these type of capillaries. So I have it listed as the glomeruli in the kidneys, the renal glomeruli in the kidneys, and also say small intestine, the villi of small intestine, as well as endocrine glands. So the glomeruli in the kidney, this is where filtration of your blood will take place. We're trying to get rid of waste products. And so a lot larger things are going to get through because the whole process is to try to get rid of waste products. In the small intestine, this is where digestion and absorption will take place. So I have to be able to allow certain digestive enzymes to go through. I also have to be able to absorb certain nutrients, and so this is going to allow bigger things to get through. Endocrine glands produce hormones, so I have to have a bigger size to allow these hormones to get through. Now the last type has two names. It's some refer to as being discontinuous, or I'm used to referring to them as sinusoidal capillaries. Now these type of capillaries have an incomplete basement membrane. So you see there's not complete here. Plus you have these very large gaps between the cells. These are found in things like your bone marrow, your liver, and the spleen. These type are the most permeable of all the capillaries. Now let's think about it. Why would I find it in bone marrow, liver, and spleen? So these ones will allow proteins, large proteins, and blood cells easily to get across. The other two don't. So this one is going to be allowing large proteins and blood cells, and say, well, how can correlate to where they're located? Bone marrow. This is where your white blood cells, your platelets, your red blood cells are produced. So I got to be able to have these things get across. Liver. Liver is where a lot of plasma proteins are produced, like albumins and immunoglobulins. So the liver needs to have these type of capillaries. The spleen as well as the liver are involved in the breakdown of red blood cells. The spleen filters your blood and is part of your immune system. So it's filtering blood, so we've got to have those red blood cells to be able to get through. So this type of capillary is the most permeable. We'll remind you of this when we talk about the lymphatic system, because the spleen is part of the lymphatic system, which is lymphatic/immune system. We'll talk about this again when we do digestive system, when we talk about the liver and having those type of capillaries. So we have 3 types of capillaries, all involved or in exchange, but we have differences in permeability based upon their structure. So this is where you see again that concept of complementary structure and function. Now in the capillaries, I'm just going to show you, when we look at a capillary bed, which is kind of like just a network of vessels, I want to look at some of the structures that are part of the capillary bed. Okay, here you have an arteriole, and the arteriole is bringing blood towards the capillary bed. In some place in our body, we have what we call metarterioles. So metarterioles are initial segments in the passageway between arterioles and venules. Now the metarterioles, I have to make a distinction, is that metarterioles, if you kind of look really carefully, you can see these little dots here, is the metarterioles do not have a complete smooth muscle layer around it. Arterioles do. You see the smooth muscle there. They don't have complete smooth muscle around it. Now, they can act as thoroughfare channels, where if they, for example, these structures here, right before the capillaries, these are called precapillary sphincters. If those are contracted, blood is not going to go into this capillary bed. It can bypass, go through this thoroughfare channel, and go directly from the arteriole to the venule. So if I say, no, don't really need to go there, I'm just going to bypass it and go this way. So they refer to that, it can be as a thoroughfare channel. Now, precapillary sphincters are a smooth muscle that control blood flow to a capillary bed. So if the precapillary sphincters are contracted, blood is not going to go into those capillaries. But if they're relaxed, blood will. And the thing is, what are some things that could affect that blood flow to the capillary bed? And that's one of the lectures that we're going to be doing later. But I do want you to write this down, is that when we talk about the autonomic nervous system, the sympathetic nervous system can innervate blood vessels. Well, the sympathetic nervous system can innervate arterioles and affect the contractility of that smooth muscle, which could affect the diameter of that blood vessel, which could therefore affect blood flow to the capillary beds. However, there is no innervation by the sympathetic nervous system to metarterioles or precapillary sphincters. So, what things are going to affect whether or not that smooth muscle is contracted or relaxed? This is where we're going to talk about at a later time, is about local factors that can affect blood flow. Things that can affect the contractility of that smooth muscle. Things like carbon dioxide and lactic acid. So the autonomic nervous system won't affect these, but it will affect the arterioles. So I do want to mention that. Now another thing I do want to mention is this term called collaterals. Here, this picture, you can see an example of it. This is an arteriovenous anastomosis. So anastomosis is a fusion of 2 vessels, and here it's between an arteriole and a venule, hence the term arteriovenous anastomosis. They do exist in our bodies. So in AV, we call it AV anastomosis. That's that direct connection between the arteriole and venule. That will allow blood to bypass the capillary bed and flow directly into the venous system if that anastomosis is dilated. Now we do have what was referred to as collaterals, and technically an AV anastomosis is an example, which you see here, of technically a collateral, but I want to talk about specifically collaterals that open up. So a collateral, where we say collaterals in general, are we have multiple ways that we can supply blood to an area. Think of them as backups. So we all have collaterals to some extent, but you don't really see it here. They're microscopic. But what's going to happen is if you see at B, where we're starting to kind of block blood flow. So this is someone's maybe developing atherosclerosis, some plaquing in that vessel, and that's going to affect the amount of blood getting through that vessel to up this tissue up here. And so you'll notice these are collaterals right here. They start opening up, and actually we can start developing even more collaterals as this starts to notice there's a complete blockage here, but blood is getting to where it needs to through those collaterals. It's kind of a backup. If someone has well-developed collateral circulation, they could have a complete blockage in a coronary artery and never have a heart attack, believe it or not. People who've had heart attacks, those who develop collateral. Now I apologize, I got a call, and so I got distracted, so I don't know exactly where I ended. So I just want to mention that the collaterals are just, again, a alternate way of getting blood to an area up in the body. We'll just leave it at that. So far we've got the blood going from the arteries to the arterioles to the capillaries. Talking about the capillary bed, which you see right here. And you notice here you're seeing that blood going right through that thoroughfare channel because these precapillary sphincters are contracted. I'm going to stop here because I'm trying to keep recordings relatively short, like 20 minutes or so. We'll continue on looking at the veins, the venules and the veins, and talk about venous blood flow as well as blood distribution through the body.